could have questions now. The microphones in the center aisle. And tell us your name and location.
Bob Orsh from Birmingham. What's the lifespan of the flying moth? I'm curious.
Even in the wild, as adults, their primary purpose is dispersion of the genetic material. And so they tend to last for maybe a week or two. And so they spend most of the time as caterpillars and as cocoons. And once they emerge, their task is to find, if it's a male, it'll try to find the female. And as you know, they have extremely sensitive chemoreceptors.
My wife says the same thing about me, by the way. A week or two.
Do they, can you change their behavior? Is that part of the scheme?
Yeah, so the intent of those experiments was to stimulate them to cause a change in the flight path. And so we would give them a target, like a flower that they can track.
Right.
Because in the wild, they go around sipping nectar from flowers. And so they tend to fly towards flowers. And in fact, in the lab, we have a robotically controlled flower, so you can program three-dimensional trajectories to these flowers. And as the moths try to fly towards them, we can stimulate to cause them to veer from the flight path. But as they try to re-zoom in on the target, we can study how they're responding or how they are using their flight muscles in order to do that. And so you can do a control system analysis. And the Office of Naval Research was interested in that because they wanted to make artificial flapping, flying vehicles that are unmanned or manned and that can track targets or whatever.
Well, I see. So the main purpose is to analyze their flight.
Yes, to analyze the flight, yeah.
Please.
Chris Phoenix, Center for Responsible Nanotechnology. So obviously you could fit hundreds, if not thousands, of those flight chips into one of your cans. Have you thought about what investigations you can do if you're recording 1,000 neurons and correlating them, maybe even from different parts of the brain? How close are you to figuring out how brain circuits work as opposed to just how neurons work?
I think there are several research groups attacking that same problem of trying to increase the number of channels in the system and doing lots of correlations such as that. Even at UW, there are groups interested in that. And they've been, well, there's also interest in looking at local field potentials and ECOGs, for example, because it is the activity of a population of neurons that's of interest in many cases, and not necessarily from a single neuron to a single muscle.
Hi, Linda McDonald-Glenn from the Alden March Bioethics Institute. Most of the research I've heard here has been directed towards telemonitoring, and that's great. I was just wondering, though, if there was any research that was geared towards, especially with the animal models, if there was any research towards augmented cognition with biochips. If either of you were aware, if any of you were aware of that type of research being done yet. Augmentation with microchips or biochips. No?
Well, I've heard, at least people mentioning the passing, like retinal implants, for example. You could easily alter your sensor's characteristics towards a UV or, using, or towards ultraviolet or infrared, and that might add additional capabilities to the person who's implanted with it. But I'm not sure if anyone's seriously considered augmentation in other directions.
With animal models yet?
With that, yeah.
Okay.
And the same goes for cochlear implants too. You could alter your system's properties to give the person new hearing capabilities.
Okay, thanks.
Just a brief comment. I don't know what counts as augmentation, but BCI work is arguably augmentation. So the brain-computer interface work. So yeah, there's a lot of that happening.
Yeah, that's a good point. Yes.
Any further questions? Any additional comments from the group here? I suggest that we take a few minutes just to walk around. We want to make sure we're as alert as possible for the last three speakers. So why don't we take, what, 5, 7 minutes?